It's now my pleasure to invite Maddie James from down the hallway. Well, I'll allow you to introduce your talk. Thank you very much.
Wonderful. So hi, I'm Maddie. I'm a postdoc in Daniel Ortiz-Barrientos' lab, and our work is all about uncovering the genetic basis of adaptation and speciation. And we work on Australian native wildflower called Nesio. So today I'll be giving you a broad overview of the type of work that we do in our lab. So I'd first like to begin by thanking the wonderful team of people that have contributed to the work over the years. Obviously, without them, none of this research would be possible. So a major goal that many of us have that in the centre is that we want to link the genotype, so the genes, to the phenotype, or the traits, to the fitness or the success of plants. So in other words, we want to understand the genetic basis of adaptation. And understanding this is super important. On one hand, it can help us create elite crop varieties by knowing which genes to select to increase crop yield. On the other hand, if we look at natural systems, knowing the genetic basis of adaptation will help us understand how plants adapt to harsh environments and whether different species can solve environmental challenges in similar ways. And this is exactly what we are interested in, understanding the genetic basis of adaptation in nature. So when we talk about the genetic basis of a trait, what do we actually mean? So let's say we have a plant trait, which is leaf shape. The genetic basis of it not only encompasses the single DNA changes and genes that underlie the trait, but how much each gene contributes to the trait, how these genes are regulated and expressed, how they come together to form larger pathways and gene networks, patterns of methylation across the genome, how other features of the genome, such as structural variation, impact how traits evolve over time, and how this is all modulated by how the DNA is packaged and coiled into the 3D genome structure. And so this is exactly what we're trying to uncover in the system that we work on, but not just for any traits, but those traits that are actually beneficial and facilitate adaptation in nature. So let me tell you about the system that we work on. We work on an Australian native wildflower, the Nesio lauda species complex, and it contains many varieties, or what we call ecotypes, that are distributed all across Australia. So here you can see the numerous ecotypes. Each has genetically based morphological differences, and each ecotype has adapted to a different environment. For instance, we have ones found in the sand dunes, which grow tall, and ones in the rocky headlands, which are short. And so we want to understand how natural selection has driven the evolution of these ecotypes. So just to recap, if we want to understand the genetic basis of adaptation, we need to understand variation and diversity at all of these levels. However, it's actually quite difficult to make causal links between the underlying genetic basis and a trait, or specifically an adaptive trait in natural systems. So let me tell you why. So let's say we have a plant population that has adapted to a new environment, and in doing so has evolved a new morphology. You might first be interested in how this adaptation took place, so you might first want to examine which traits increase fitness in the new environment. So in other words, which traits are adaptive. But just because a trait differs between the environments does not necessarily mean it's involved in adaptation. It might just be correlated with other adaptive traits. So you can't actually tell which traits are involved in adaptation just by comparing the two populations. Well, then you might go on and do some population genomics approaches to look for signatures of selection in the genome to ask which genes are under selection. But again, just because variation in a gene is different between the populations does not necessarily mean the gene is involved in adaptation. Then, on top of this, you do not know which genes underlie which trait. So how do we deal with these problems? But fortunately, these issues can be solved with a touch of magic. Or in other words, a magic population, which is a multi-parent advanced generation intercross population. So I'll show you what it is. So magic populations, they're constructed by crossing populations repeatedly over many generations. And this is what we've done in Nesio, where we've crossed many individuals from a tall dune population with many individuals from a short headland population for 11 generations to create the magic population. And so this magic population is all mixed together and shuffled up. So those two original founder dune and headland populations that come from one location along the Australian coastline, those founders have been deconstructed. And so this allows us to link the genotype, phenotype, and fitness by breaking down trait correlations. So I'll show you how it does this. So let's say we have a chromosome from the dunes and a chromosome from the headlands. In the dunes, let's say we have a region of the genome associated with being tall and another region with having large leaves. In the headlands, these regions are associated with being short and having small leaves. So tall plants will always have large leaves and short plants will always have small leaves, which makes it really difficult to look at the genetic basis of these traits in isolation because they're correlated. However, in individuals in our magic population that have undergone many generations of crossing and recombination, we've been able to deconstruct this trait correlation because tall plants do not always have large leaves and short plants do not always have small leaves. So now we can link the genotype to the phenotype with this. But how do we know then what traits are actually adaptive? And we've been using the Nesio system in a really unique way to answer this. So what we've done is we've taken our magic population that's all deconstructed and shuffled up, and we've planted it back into the field, into those original contrasting dune and headland environments. And this allows us to see how natural selection favors and reconstructs those favorable trait and gene combinations in each environment. So to do this, we glue seeds onto toothpicks and we plant them in many of these replicate grids across both of the environments. And this allows us to track the identity, growth, and survival of each seedling. So the best surviving individuals, or those that are fittest, we've taken them, crossed them, and then they've gone back into the lab for a total of 3 rounds of selection. We can examine then the survivors in each environment to see what natural selection has favored in the wild. And these transplant experiments in the system have enabled us to identify key adaptive traits, one of them being gravitropism. So gravitropism, I won't go into too much detail, but it's the ability of a plant to respond or bend to that downward pull of gravity. And our research has shown that it's correlated with those overall growth habits in Nesio. So this is where tall dune plants do respond to gravity, whereas short headland plants do not respond to gravity. So now that we have our adaptive trait, we've been using whole genome sequencing in individuals of that magic population to uncover the genetic basis of it. So we have a genome-wide association study. You've seen a few of these already in the previous talks. Along the x-axis, we have our chromosomes, and effectively any dot that lies above that dashed line is a region of the genome that's associated with gravitropism. So what we're finding is that adaptation is complex. It's not just one or a few genes that seem to be contributing to this adaptive trait, but many genes distributed across the whole genome. For instance, if we zoom in on chromosome 17, we can see a few strong peaks here that are highly associated with gravitropism. Now, what is really interesting is that we're also finding that some of these gene regions, these candidate gene regions, also reside in chromosomal inversions. So we have these co-adapted gene complexes that are trapped together because of the low recombination in inversions. We are also combining these results with population genetics approaches in those original founding dune and headland populations to ask whether natural selection can, or confirm that natural selection is actually driving the evolution of these gene regions. We are also looking at all of this here in another 30 traits or so and examining how natural selection simultaneously acts upon multiple traits. So these multi-trait responses to selection. And you'll hear more about this in the talk from Candace tomorrow and looking at, how natural selection can simultaneously actually, evolve multiple traits at the same time. Okay, so at this point of the talk, you might be thinking, so you guys have created a magic population that come from one dune and headland population at one location along the coast of Australia. but will adaptation always proceed in similar ways? So in other words, if we went back in time and replayed the tape of life, would we still see the same kind of genetic solutions when species face similar environmental challenges? Now, there's something really interesting and useful about this Nesio system that I didn't tell you about before that allows us to answer this question. We have replicates of evolution, so you might have heard of it from Barbara, what replicates are. So effectively, the dune and headland ecotypes don't just occur at one location along the Australian coastline, but they've independently adapted to many locations. So you can see all along the Australian coastline, we have these dune and headland contrasting environments that harbour the dune and headland ecotypes. And we have really strong evidence to show that each population is an independent replicate of the evolutionary process. So with these replicates, we've been asking how repeatable evolution is at different levels of biological organisation, so the SNP, gene, genetic pathway, and network. And we are finding that each replicate population has adapted by a different genetic route. So each replicate is using a different set of alleles and genes to produce the same phenotype. However, there seems to be more repeatability at the level of the genetic pathway. So even though each population is using different alleles and genes, these belong to the same genetic pathways. So there seems to be more evolutionary constraint at the level of the pathway. So all of this work we're doing in the lab is kind of anchored in evolutionary theory, and we are testing long-standing fundamental models of how adaptation proceeds in nature. For instance, we're asking whether structural variations such as chromosomal inversions can facilitate adaptation in the face of gene flow. We're looking at whether gene expression and gene networks and how they can kind of underlie adaptive strategies in nature. We are looking at how genetic architectures evolve over time and examining how parameters such as mutation, recombination, and genetic drift can impact how natural selection kind of impacts the rate of adaptation over time. And we are also looking at whether the adaptation process can result in reproductive incompatibilities between populations and drive the evolution of new species. So to do all of this and more, what we've been doing is we've been constructing a Nesio pan-genome, identifying chromosomal inversions, exploring the 3D genome structure. We've measured gene expression during gravitropic responses in that F11 magic population. We're undertaking forward population genetic simulations in SLIM, and we've measured many components of reproductive isolation across many of these populations across Australia. So overall, our work in Nesio is enabling us to link the genotype, phenotype, and fitness in a natural system. We are understanding the adaptation of plants to harsh environments across Australia. Our natural replicates of evolution are allowing us to look at how repeatable evolution is at different levels of biological organisation. We can also ask whether the genetic information from Nesio can be transferred to other species. And finally, the work we are doing in the lab with the natural replicate populations and the magic population will help us create models so we can predict adaptive trajectories into the future. And this is really crucial to understand how organisms will adapt in the face of climate change. And with that, again, I'd like to thank everybody in the group, and you'll hear more from us in the upcoming days. Thank you.
That was fantastic, wasn't it? Very clear and beautiful story. Wonderful. Thank you so much. Any questions? Peter's going to ask another question.
Asking. So you said that each of these different pairs is an independent evolution. So how do you know that, and how long have they been evolving to actually create these differences?
Yes, so I wrote a paper on that, so very good question. Effectively, well, there could have been a scenario where like the dune populations just arose once and then like traveled along the coastline, and then the headlands just once and traveled along the coastline. So the phylogeny, we see our replicate dune headland pairs as sister taxa, and there's also like really little gene flow between them. So the gene flow like between the populations aren't enough to obscure the phylogenetic signal of like, yeah, that it looks like replicated evolution when it actually isn't. And then we've confirmed this with some simulations as well. I mean, I can chat to you in more detail.
Didn't evolve once into this pair, or it just came from inland, if you like, coming up and spreading out?
Yeah, so in the phylogeny you would see dunes clustering all together and then headlands clustering all together if it was a single origin scenario. But we see like dune headland, dune headland, dune headland, like the eucalyptus wet-dry contrast that Barbara showed us before, if that makes sense. Divergence times, do you mean between the dune and headland of each pair? Yeah, some of them, the most recent ones is like tens of thousands of years. So they're quite, it's a system of rapid evolution as well, so it's quite recent. Not millions for the dune headland, but more millions for the entire species complex. Yeah.
We call that local adaptive evolution, is that right?
Yeah, rapid evolution.
Rapid evolution.
Yeah.
I've got the microphone. Yeah, that was great. Very clear talk, Maddie. I was just wondering, with your selection is always based on mortality, is it? From when you're doing your planting out, I wonder if would you get different results if you were looking at sort of fitness in terms of reproductive success?
So the fitness components for the field selection, it takes into account like the germination of the seed, ability to pass 10 leaves and something else. Everybody's nodding over there. So it's kind of like, it's an Astor model, so it's like conditional on passing through a variety of like life history stages to get to that fitness value. Because of course, I mean, you can measure fitness at various stages, so it's kind of contingent on the organism passing through multiple of those stages. Yeah.
Okay. Call it quits. Thank you,
Wonderful.
Maddie.
Thank you.
Very impressive.